All right, guys, today we're going to talk about meiosis, which is this word right here. Now, meiosis sounds a lot like the word, remember, mitosis. And mitosis was a type of cell division where I make new body cells. Remember, body cells are also known as somatic cells. But we have another type of cell in our body. Those are going to be sex cells. So mitosis makes body cells, but my sex cells, like think egg and sperm, that's what makes me. So meiosis or meiosis is the type of cell division that creates my sex cells, which are either egg or sperm. Okay, so egg or sperm, that's how you were made. We're by egg and sperm. So this type of cell division, I'm going to create those two types of cells. Now we're still going to go through the cell cycle. And remember, cell cycle is made up of these three parts. We have interphase, where I double my contents. And don't forget, contents, I'm talking about my DNA, and I'm talking about my organelles. Okay, I want to double everything. Remember, I want to double everything so that way when I split it, I have the right amount to function. But now, instead of going through mitosis, we're going to go through meiosis, because we're going to make our egg and our sperm today. And meiosis, by definition, is division of the DNA. Okay, I'm going to divide my nuclear contents. And then my last stage, cytokinesis. Remember, cytocell kinesis is movement. I'm going to divide that cytoplasm. And cytoplasm is including all of those organelles right there. Now, this is a lot of vocab, so we need to get through it. Meiosis, I'm using sex cells, which are also known as germ cells. These germ cells are going to be found in gonads. And I know that whenever you guys usually hear the word gonads, you think that you're referring to boys, you're thinking you're referring to testes, but really gonads, from a scientific definition, is the organ in which you make your reproductive cells. So testes would be gonads, but so would ovaries, because we have to make both of them. We have to make egg, and we're going to have to make sperm, especially if you want to have fertilization and create new offspring. So meiosis, sex cells, germ cells, these germ cells are found within your gonads, okay? And depending on what kind of gonad you have, if you have testes or if you have ovaries, then you're going to make something called a gamete. Gamete is like a big word. It's just like gonad. Say gonad can refer to ovaries or testes. Gametes can refer to either egg or sperm, depending on which one you have. Okay. So now, one difference between meiosis and mitosis is that meiosis, I get to divide twice. And so if I look, remember, this is going to be my parent cell right here. In my parent cell, I can divide it, and I can get two cells. See, 1, 2. That was one round of division. But meiosis actually divides twice. So that means in the end of this sucker, I'm going to end up with 4 cells. And we'll talk about why that's important here after a little bit. But we need to know that meiosis divides twice. So I'm going to go through a phase called meiosis 1, and then I'm going to go through a phase called meiosis 2. And it's the same stages that we've learned about before. I'm still going to go through PMAT. Remember, prophase, metaphase, anaphase, telophase. But now I get to do PMAT 1, with meiosis 1. And then meiosis 2, I get to do PMAT 2. Okay? Now, a few more words that we have to be aware of. When we went through mitosis, we talked about chromosomes and replicated chromosomes, but now we have a new thing called homologous chromosomes. Okay? Now, homo, let's think about word roots. Homo means same. And then chromosomes hold genetic information. So homologous chromosomes are chromosomes that have the same genetic information on them. All right, so these two right here, these two X's are homologous chromosomes, because maybe like right here at the end, maybe that's where eye color is, right? And so since they both have eye color at that spot, and maybe right here on this leg, or on this spot, we have height, and maybe height is right here. Since each chromosome is coding for the same information, then they're known as homologous chromosomes. They code for the same stuff. And one question is like, why do you have two of these? Well, it makes sense because you're going to get one from mom, and you're also going to get one from dad. Okay, say you guys were created by sex. Your parents have egg and sperm, and then when they came together, they gave you actually two copies of every chromosome. Now, because I have these chromosomes right here, these two X's, I have 4 chromatids. Because remember, those are the sticks. So I have 1, 2, 3, 4. I have 4 chromatids. That's going to be a tetrad because tetra means 4. And then synapsis, that's just going to hold that tetrad together because once I found my friend, so once I found this guy, then I'm going to hold his hand so that way I don't lose him. Okay, so we talked about how meiosis has 2 rounds of division. So the first round of division, I'm going to divide these homologs. And homolog is just a fancy word for homologous chromosome. So that's what I'm going to do in the first round, is divide those apart from each other. And then the second round of division, meiosis 2, this is when I divide those replicated chromosomes like we had in the past, like when we did for mitosis. Now, one process that we're going to see during meiosis is something called crossing over. Okay? Now, crossing over by definition is the trading of genetic material between homologous chromosomes. So look right here. I have a blue X and I have a green X. And they each code for the same information. I just used different colors so you could see it a little bit easier. But maybe this leg right here and this leg right here, they trade places. And so now I have a big blue X with a green leg and a big green X with a blue leg, right? Say it still codes for the same information, but now they're in a different order. So that means I'm going to increase genetic diversity, okay? Every time I do crossing over, I get to increase genetic diversity, which is really, really good. This is why if you look at a family, and you have brothers and sisters from the same parents, they're not identical. Like they share similar traits and characteristics, but they are also unique, and they're unique because of this crossing over right here, because I can switch places with my homologous chromosomes and get different varieties. Okay, so let's draw this sucker out. All right, first, I always have to start with my parent cell. Okay, so here's my cell, here's my nucleus, and then I have 4 chromosomes in there. Okay, I have 4 of them. 1, 2, 3, 4. I have a big yellow and a big blue and a little yellow and a little blue. Okay, so the big yellow and the big blue, those are homologs, and the little yellow and the little blue. And remember, you get 2 of every chromosome because you get 1 from mom, you get 1 from dad. And now that I'm going to go through cell cycle, first I'm going to go through interphase. I'm not going to go through and draw the G1, SG2, but remember during interphase, I double my DNA. So now instead of having 1 big blue stick, I have 2 big blue sticks. Instead of having 1 big yellow stick, I have 2 big yellow sticks, and then so on and so forth. So now I have replicated chromosomes of the chromosomes I started out with before. All right, so I double my DNA. Sweet. So now I get to go through my first round of division, which is meiosis 1. And now that I'm in meiosis 1, I get to start with prophase 1. And everything that you guys learned about prophase with mitosis is still pretty accurate. Like the first thing, my nucleus is going to disappear. So I'm going to start cutting holes in this nucleus. That way my chromosomes can move away or move around. The chromatin condenses into chromosomes. Now, I didn't show that because I was trying to make this a little bit simpler, but remember, my DNA is usually all stringy-like, and then it's going to condense into these nice, easy-to-move replicated chromosomes. Now, one thing that's super special about prophase 1, there's 2 things, is that my homologs pair up. So see, look, you can look right here. My big yellow and my big blue X pair up, and my little yellow and my little blue X pair up, right? Those are homologs. And then once they pair up, I can go through crossing over. So if you notice, my big yellow X has a little bit of blue, and my big blue X has a little bit of yellow. And then same thing for my little X's. Little X has a blue, little blue X has a yellow, right? So I have crossing over. I just increase genetic diversity by doing that. And then, of course, the rest of the stuff happens. Centrioles migrate, spindles form. All right, now I get to go on to metaphase 1. And remember, meta means they line up in the middle or your equator. But now, instead of your chromosomes lining up in the equator, I have my homologs. Anytime you see the word homologs, this is meiosis 1. So over here, they paired up in prophase 1. Now they line up in the middle. And you can see here's my centrioles at the edges. They're forming spindle fibers. Those spindle fibers attached to the centromere. And they're getting ready to pull them apart. But those homologs are paired up in the middle. Then anaphase. So now these spindles, remember, they start to shorten. And when they start to shorten, that's going to pull these homologs apart. So that one's going to go this way. This one's going to go that way. That one's going to go that way. That one's going to go that way. They're getting pulled apart. But instead of it being replicated chromosomes getting pulled apart, I'm separating my homologs. Then I get to go to telophase 1. In telophase 1, it's exactly like telophase that we've always talked about, how our nucleus starts to come back. And I get a cleavage furrow if I'm an animal cell and a cell plate if I'm a plant cell. But that's all I did in meiosis 1. You guys can see. I'll try to get it all on one page. There's prophase. They condense and pair up and cross over. Metaphase, they in the middle. Anaphase, they pull apart. Telophase, I get a nucleus and my cell starts to split. So now, once I go through cytokinesis, I'm immediately in meiosis 2. So now I jump into prophase 2. In meiosis 2, this looks identical as far as structures go, identical to mitosis. But now instead of having one cell, I have 2 cells because I just divided. So prophase 1, my nucleus starts to disappear. So see, like I can kind of, whoa, I got cut a little bit too much. I can start cutting a little bit of my nucleus like that. And my centrioles move to the poles. In metaphase 2, instead of homologs lining up in the middle, now I just have my replicated chromosomes lining up in the middle of the equator and the spindle fibers attached. So see, look, spindle fibers, they're attached to those centromeres. After metaphase 2, I go to anaphase 2, which means my spindles start to shorten. Oh, didn't do it over here. There you go. So my spindles start to shorten. And when they shorten, that pulls those replicated chromosomes apart, right? So one goes this way, one goes this way. One goes this way, one goes this way, and vice versa over here. One goes this way, one goes this way. One goes this way, one goes this way. I separate them. Then in telophase 2, again, I just undo everything. So now my nucleus starts to come back, and I start to have a cleavage furrow, and my chromosomes will condense back into chromatin. Then after cytokinesis, you guys can see that I have 1, 2, 3, 4. I have 4 cells, right? And these 4 cells are all, if you look, so like, look, I have a big yellow and a little blue, or big yellow, blue, and a little yellow. In number 2, I have a big yellow, and then a little bit of blue, a little bit of yellow. And then 3 and 4, you can continue on. Are any of these cells identical to each other? Do any of them have the same pattern? No. These things are all genetically different from each other. Okay, so that's the big thing, genetically different. That's because of crossing over. Also, each of these cells have half the material I started with. So if I go way back up here to parent cell, remember parent cell, I had 4 sticks in there. I had 2 big and I had 2 little. Now, all the way down here, I have 1 big stick and 1 little stick. So I have half the information, okay? Which is what I want to do. Because, vocab word, having half the info, this is called haploid. and then having the full amount, this is called diploid, which is easy because di means two, and I get one from mom, I get one from dad. Now, these cells right here, okay, these are my gametes. Don't forget, gametes is just a fancy word for egg and sperm, okay? I just made either 4 egg cells or I made 4 sperm cells. Now, you want these egg and sperms to have half the information in them because when they come together, they're going to make a full cell. So here's my egg, right? It has a big stick and a little stick. My sperm has a big stick and a little stick. Those are haploid. And then when fertilization occurs, when they come together, then I have a fertilized egg, which is called a zygote. And now look, now I have 2 big sticks and 2 little sticks, right? Because fertilization restores that diploid number. Then this little sucker can go through mitosis, and then mitosis, because remember mitosis is about growing your organism, then he will turn into a full organism, okay? There you go. Okay, so let's go back through it really quick. So there's your parent cell, 2 big sticks, 2 little sticks. DNA doubles, so now I get my X's. Prophase 1, they pair up and they cross over. And then entirety of meiosis 1 is about dividing these homologs. So there they are in the middle. Now they're being pulled apart. And now I have 1 in each new nuclei. Then in meiosis 2, I have 2 cells, and I'm going to divide up the replicated chromosomes. So nucleus disappears. I put the replicated chromosomes in the middle. I pull my replicated chromosomes apart. I form 4 new nuclei. I go through cytokinesis. I make 1, 2, 3, 4 cells, all genetically different from each other, which is what I want, because genetic diversity will allow an organism to have a better chance of survival, perhaps. And since they have half the information, they're called haploid, which is a good thing, because if each of my gametes is haploid, then when they come together, they will restore that diploid number, because you get 1 from mom, 1 from dad. Okay, I hope that helps.